Hello, guys. Good morning or good night. This is morning here in Vietnam. So right now I will present my research paper, which is a lot of effort for me. More about me later, but yeah. This is the title. As you can see, I have analyzed some key term that you don't need to afraid about it. Like if you have no biology background in the last few months like me, this is words quite imitating, but I have been able to do this. So I would try to explain to you all really simple about all these terms and like how is this matter. So I will say it again. My research title is like comparative analyst of program reversal frame shipping in Chikungunya virus strain from Africa, Asian, and the Caribbean using amphiphil-3 and TRORS RNA. So first, we will want to know what is this chikungunya virus. So, as you can see here, it's a mosquito-borne virus that are typically endemic in African and Central Asians, and it is also spread widely with its transmission documented in Europe, Australia, the Pacific Island, and even America. And because it is transmitted by a mosquito, is really prevalent in the Southeast Asian, especially in my country, Vietnam, in the rainy season where this mosquito is kind of favor their reproduction. So, some symptoms of this chikungunya virus when you contract the disease is that you have kind of fever, red, and severe joint pain. That is kind of lenient, but severe neurological outcome had also been reported. And although more patients recover within 2 weeks of, but some also develop long-term arthritic complication, and that can result for months or years. So it is quite troublesome for the others. But the most serious thing about this chikungunya virus is that there is currently no FDA-approved antivirus or vaccine for this. So like when you contract the disease, you can only be given medicine so that it's alleviate the pain, but not cure you from the disease. Yeah. So what is program reversal frame shipping? So you can see the picture taken from Wikipedia here. This is like the biological process where the ribosomal ship is reading frame by one page during the translation. So it's like you have one recipe, but you have kind of many dishes. Like from this, you can kind of create many dishes. So that is kind of what this mechanism do. So it's allow a single messenger RNA to produce multiple unit protein from overlapping open reading frames. So this mechanism is what this virus deploy to kind of reproduce. So here is the 2 strain I taken for the research. The first one is African Asian, and the second one is the Caribbean. So you can say they kind of have really similar sequence alignment. However, there's one difference you can see here with the red arrow. So that different, although really minor, like A and C, but it will have kind of different, like you can see 2 image here, this kind of different. And when you kind of bring it on to the software and read it is. So that is where my research closely about. So next slide. Here is how I do it. So first, I take the sequence from the paper called Functional and Structural Characterization of the Chikungunya virus translational recording signal from the author. Big thank you to them all. So when I take these like sequence, I will put them on for structure prediction with Rosetta RNA and amphiphil-3, and then I download them and I put them on Chimera X for structure alignment and visualization. So here is the result. So first, I would try to visualize the Caribbean strain. You can see here is like where I'm pointing here is the Caribbean strain generated with M443. And this is what it is also the same strain but generated through the TR Rosetta RNA. So you can see the big difference here. Why this video had kind of a really well-defined loop and turn, as you can see here. It's really kind of a parallel helical stem with, stem look helix, really well-defined. But on the other hand, the same sequence put onto the Rosetta RNA is a kind of messier, with more open loop. You can see here, like this part here, more open loop with fold and bend, yeah, here. So it's have bend helix and extended loop, so that you can see more clearly here when I align these two together to kind of know the big difference. Yeah, although they have kind of this really similar reason, like the backbone hyperloop really converse, they kind of have the bit different here and here, where the tyrosine RNA the image generated by this app is like more extended. So it's quite obvious. And the same thing happened when I do the research with the African Asian strain. Again, really well-defined stem loop helical. You can see here by M443. But on the other hand, Rosetta RNA, kind of more messy. Again, although it do have kind of well-defined stem loop here, it's have open end and kind of messy loop here. So you can see the difference, but more of it here. Again, the backbone, the helical stem loop here, they have the similar things, but this is where the difference begins. They stand more clearly the model from TL Rosetta RNA. So what if I compare the Caribbean and African Asian strain using the different app? So I'll start with M43 here. Really similar one, although some deviation you can see here or like here is visible, but they are kind of compact stem loop helical and kind of similar. But on the other hand, you can see TL Rosetta, they generated kind of really different one. So yeah, we can still see the similar, but the African Asians stand have like this extended look and like really messy part here where they're not really similar. So yeah, that is the whole end of this. So what all of this mean? This means we currently there is a limitation for the current model. M443 would favor stable fold, while Rosetta RNA might capture kind of unstable but maybe biological relevant state. Yeah, and this is maybe indicate a possible subtle lineage-specific effect on the RNA folding that warrant further investigation. And in conclusion, like just to recap you all so you can kind of catch it. M443 predict a nearly similar structure between the two strands, while the oral state that RNA so widely different. And why M443 predict a more compact stem locality, the other app predict an alternative with more flexibility and open loop. This highlight both the promise and limitations for computational RNA modeling and point out the need for more experiential validation. So this is like a gap of knowledge. And also this RNA, when we kind of tackle this, can be targeted to develop novel antiviral therapeutic approach with maybe kind of help with the current problem when you have like no approved way to kind of cure the disease. So yeah, lastly, I want to give a big thank you to my mentor. He has helped me a lot, really patient, really helpful for last month. And also big thank you to the Talaria Summer Institute for giving me this wonderful opportunity to kind of present my research, doing it, and kind of step out of my comfort zone. And lastly, thank you all for listening.